Let's talk about protein synthesis, which is also termed as translation. The mechanism of protein synthesis has got other name, that is translation. What exactly happens in protein synthesis? So in protein synthesis, the amino acids are joined with each other. Say this is first amino acid, this is second amino acid, this is third amino acid. So the formation of the chain of amino acids in a particular in a specific series is referred as protein synthesis or translation. So we know that for any amino acid, there will be two important groups, two important functional groups will be there. That is the NH2 group, which is amino group, which is the amino group, and each amino acid will also have a COOH group, which is called as acid group or acidic group. Okay, hence an amino acid, hence the amino acid has got its name as amino and acid, amino acid. So because any amino acid, if you take, it will have two important functional groups, that is, one is the amino group and the second one is the acidic group. Now what exactly happens during the protein synthesis is, say this is our first amino acid. Similarly, the second amino acid, which also has a amino group and a carboxylic acid group, right? So the formation of a bond between this COOH group of first amino acid and the amino group of the second amino acid, a bond is synthesized between these two groups, which joins the first amino acid with the second amino acid. Similarly, the second amino acid will be joined to the third amino acid with the same kind of bond formation, right? So this process of joining of one amino acid to other amino acid and making a long protein chain of amino acids is generally referred as translation. To talk more about translation or protein synthesis, it is important to understand the functions of three components in a cell. The first component is mRNA. We should talk about three components. The first component is mRNA, the second component is tRNA, and the third component is ribosomes. Because when we talk about this whole protein synthesis, these three components will come into the real action. What is mRNA? mRNA is actually synthesized basing on DNA, and this mRNA carries the genetic information from DNA in the form of triplet codons. So we know that the three nucleotides on the mRNA, say for example AUG, GUG, GAG, so on, they specify these, we call them as codons. Each of these triplet codon will specify a particular amino acid. For example, if there is AUG in the mRNA, it is going to specify or it is going to code for methionine. Similarly, each codon, it specifies a particular amino acid. So in this way, the information, the genetic information from the DNA is carried by the mRNA. So we should understand that the sequence of nucleotides or the sequence of codons in the mRNA is going to specify which amino acid should come first, which amino acid should come second, which amino acid should come third, likewise for protein chain synthesis. Then comes the second important component, that is tRNA. So we know that tRNA, there are around 20 tRNAs in any particular cell, which means that 20 different amino acids are carried by 20 different classes of tRNAs. So this tRNA, the main function of tRNA is transfer. That is, it transfers a specific amino acid. Specific tRNA carries a specific amino acid to the site of protein synthesis. Okay, then comes the ribosomes. So, coming to the ribosomes, each ribosome is composed of two important what you call subunits. You call one subunit as a smaller subunit, the other one as large subunit. So we can actually see three important regions or three important locations in each of these ribosomes. We call them as three important sites, that is E site, P site and the A site. These are the three regions which are important to be discussed under this translation. E site, that is exit site from where the tRNA exits. P site is peptidyl tRNA site. A site is amino acyl tRNA site. We shall discuss more about these sites and what exactly happens in these sites when we look at the process of translation. So, this process of translation can be studied about four important headings. The first one is charging of tRNA. You can also call it as activation of tRNA. The second step is initiation. Next is elongation. Next is the termination. Pause. As we were discussing, the first important step to be understood in protein synthesis is activation of amino acid and attachment of tRNA, or this is also called as charging of tRNA. So this is nothing but, as I told you, each tRNA is specific for a particular amino acid. So in this stage, what happens is the tRNA is attached to the amino acid, or a particular amino acid, a specific amino acid is attached to the specific tRNA. So this happens like this. So the amino acid tRNA in the presence of ATP, the amino acid is attached to tRNA in the presence of the energy, that is ATP. This step is actually catalyzed by amino acyl tRNA synthetase enzyme. Again, here I specify, as we have 20 different tRNAs, we also have 20 different amino acyl tRNA synthetase enzymes. Means each of these synthetase enzyme is specific for a tRNA. Means the tRNA, if it is getting attached with methionine, then you have a specific amino acyl tRNA synthetase which can join the methionine amino acid with tRNA which is specific for methionine. So in this way, because we have 20 known amino acid, there are at least 20 different amino acyl tRNA synthetase enzymes. So the amino acid is attached to tRNA to form amino acyl tRNA. So such tRNA is called as charged tRNA, amino acyl tRNA. And for this bond formation between amino acid and tRNA, the ATP energy is utilized so that ATP is cleaved to form AMP and a pyrophosphate. So here you can see this is a tRNA. To the 3' end of tRNA, you can see this is the enzyme. This complete portion is the enzyme, amino acyl tRNA, amino acyl synthetase enzyme, and here is the amino acid. So the amino acid is attached to tRNA via the enzyme unit. Let me brief about the three important steps involved in protein synthesis. The first is the initiation step. Initiation involves binding of small ribosomal subunit. So I told you ribosomes will have two subunits, the small subunit and the larger subunit. Here we are talking about the small subunit to mRNA, and then it is joined by 50S subunit, that is the larger subunit. Means first of all mRNA will bind to small subunit, then the large subunit will join this small subunit which is already attached to mRNA. So generally the initiation involves all the reactions that precede, means that happens before the formation of first peptide bond. Okay, the first peptide bond between the first two amino acids of the protein. So the first peptide bond formation, this step actually involves elongation. So all the reactions that occur before, that precede, okay, that occur before the formation of our first peptide bond, they all come under the initiation phase of protein synthesis. The second stage is elongation. The elongation reaction includes the synthesis of first peptide bond and the addition of the last amino acid. Means the synthesis of first peptide bond till the last peptide bond. So whatever the reactions happens, all that comes under the stage 2, that is elongation. Then termination is the last stage, which includes all the steps that are needed to release the completed polypeptide chain. So once the polypeptide chain is synthesized, it has to get detached from the ribosome and it has to get released. So all these reactions will occur during the final stage of protein synthesis, that is termination.